Everybody's back. Welcome again, and thanks for taking a week out of your what I'm sure are busy experimental schedules and coming here. And so I'm gonna give a little bit of an introduction about why modeling is useful to you, and from an immunologic background. Some examples of how modeling has advanced the fields of experimental immunology, and in particular, studies of some viral diseases. I'm going to give you some examples from the work that goes on in my laboratory about how we've used and constructed models to create representations that address both basic science and translational clinical issues. My other hat, I'm a transplant immunologist and nephrologist, a medical kidney doctor, so I take care of kidney transplant patients, and that's been the motivating factor for one of the studies that we have going on in the laboratory. Okay. So let's talk about modeling. This should be no surprise to all of you, and the fact that you're here means that you recognize that modeling is something that we do or that we would like to do. The reality is that immunologists have been creating models ever since there has been immunology. This is a picture from a talk that Paul Ehrlich, who received the Nobel Prize for his work, in actually one of the first conceptual representations of antibodies, which he called antitoxins at the time. And you can see that we have a visual model of what he called toxin and antitoxin at the time, and which most of us would recognize as a receptor ligand or antibody antigen model. And this quote from Ehrlich demonstrates one of the key issues in modeling that he says at the very beginning of my theoretical work in immunity, I made it my first task to introduce measures and figures into the investigations regarding relations existing between toxin and antitoxin. And then he gives us the punchline, which is true today as it was back then. From the outset, it was clear that the difficulties to come were extremely great. And I think what he meant was that quantifying things in biology is not easy, but you need that quantitation for modeling. So what's a model? Well, some definitions include something bigger or in general smaller than the original thing. What do I mean by bigger? Well, one of the first biologic models to have relevance for the modern era was the model of the double helix. Of course, it wasn't the dimension of a chemical piece of DNA. So in dimension, the model was larger than the thing that it was modeling, but it was also a reduction in terms of what was modeled, in terms of the degree of accuracy. So in general, models are a substitution for the original thing. They're a map. So maps are useful because you don't have to carry the entire geography around in your pocket or on your iPad. They're reductions. They leave out a lot of features. Maps are also adapted to what your question is. So you don't carry a topological map around in the middle of a city, but when you're out in the wilderness, it helps to know how high things are. All models are, in general, reductionistic explanations. It means that we leave things out and we include things that are relevant, and deciding what those things are is really one of the keys. So why use a model instead of the real thing? Well, first of all, to test something before building it. Bridges, synthetic proteins, vaccinations, immune therapies. The models may be more revealing to us than the original system. In other words, we can play with them. And in many cases, they're safer to work with than the original. All of us would much rather work with a model of Ebola virus than be in a BCL4 lab and work with the original thing. Again, the models can be manipulated in silico or on a piece of paper where the original cannot. What other things are we interested in gaining with models? Prediction, explanation, and testing are usually the big things. We want the models to enlarge our understanding of the systems that we study. So, one of the things that's important at the outset is to recognize that models have a language of their own. So this is a representation. I'm sorry, the figure doesn't reproduce quite so well, but this is from Peter Doherty's Nobel lecture. And as you may recall, he received the Nobel Prize for his studies on the CD8 response to viral infections, in this case influenza. And you can see that he's got this pictorial diagram, which is beautiful because it includes both time as well as states of immune cells, individual agents, and a spatial and state representation of the CD8 immune response. So what are the various pieces? We've got agents, the individual things that make up the model. The CD8 T cell, the influenza virus, natural killer cells, the respiratory epithelium. We've got states, so the quiescent state, non-dividing, non-activated state of CD8 T cells, the activated state, migration, and we've got different locations. All of these things have mathematical correlates in modeling, and I'm going to show you some of that. Some other models in immunology. This is a model of T cell signaling, so we've got the signal 1, TCR ligation, this little cascade of molecular signaling events. Notice the nucleus and gene transcription is not really represented very well in this model. Of course, it's an abstraction. The various cell types that are involved. So if you were going to construct a computational model of this, then you would need to know a lot about signaling and networks and how those interactions work and what the quantities involved are and who binds to what. State space models, these are models where you actually try and model the entirety of everything. In some sense. So they seek to describe the entire system in terms of inputs and outputs, and these models depend on agents, states, and relational rules which govern the changes of states. In other words, a CD8 T cell becomes activated when it sees antigen in the context of MHC class II with co-stimulation. You can make up a set of semantic or computational rules that describe those models, and on the day that you do agent-based modeling, you'll learn how to do rule-based models and to specify those things. So, types of mathematical and computational models involve the model itself, and the model is just the formalism, okay? It may be the diagram, it may be the mathematical equations, but what we're really interested in, and part of this workshop, is to give you the tools to implement that model, to do a simulation. Because really, we're experimentalists. We want to play with those things. We want to see what happens if I leave out the T cells, if I stimulate with a different cytokine, if I induce apoptosis. And we want to do it in a way that may be easier than doing it in the laboratory. So models can be dynamic or static. They're time dependent or time invariant. It depends on whether or not the time plays a role in your model. They can be discrete, meaning state variables change at particular points in time and we're not modeling a continuum, or they can be continuous. They can be deterministic. In other words, we've got a known set of inputs with a unique set of outputs, or they can be stochastic, which involves probabilistic variables and one or more random variables. By random, the statisticians mean a particular thing. It doesn't mean the variable can be anything and it's just random. It means it has a formal statistical distribution, but the exact value of the variable is confined to that particular distribution. They can be agent-based. Agents are the primary unit. We love these models as immunologists because they simulate the things that we deal with in the laboratory. A cell, a virus, the interactions between those cells. Or they can be quantity-based. These are more on the level of population models. What percentage or what number of activated B cells occur over time in the model. Or they can be hybrid, which combine two or more disparate methods of modeling. So, what are some examples of agent-based models? Now, this is not an immunologic example. I need the mouse here. This is an example of explaining a complex behavior with simple rules. It'll take me a second to get the movie up here. This is the rules for simulating flight of a flock of birds. These agents are called boids, and this was one of the very first types of simulations. And there are really four separate rules that describe this complex behavior. There is the rule of steer to avoid crowding local flock mates, so you don't run into them. There's the rule of alignment, steer towards the average heading of local flock mates. Cohesion, steer towards the average position, the centroid of your local flock mates. And attention, ignore flock mates outside of a certain distance. So it doesn't show up all that well, but you can see that here's the representation of the sphere of attention, of where those flock mates are steering towards, and you can also see that they'll avoid certain obstacles if they fall within the range of vision. These simulations, rule-based simulations with these agents and four basic rules actually simulate the complex behavior of schools of fish and birds and whatnot. And this type of simulation is used in Hollywood movies as well as in biology for certain behaviors. So is it used at all in immunology? And the answer is yes. Yeah. So this is an agent-based simulation of T cells within a lymphoid follicle, and the T cells are the blue-based cells, and you can see them migrating around and getting activated. And you're going to learn about this in the agent-based modeling. The data from these kinds of models involves spatial representation and figuring out just parameters that involve how long is a point of contact, how does a cell migrate, and involve fairly detailed experimental observations. These kinds of models are agent-based models, and they are actually fairly common. Now, this is another type of model that is agent-based and even more complex. This is work that models tuberculosis, and I know some of you are working on modeling of tuberculosis. These are agent-based models. So you've got multiple agents here. You've got T cells, you've got rules, how they respond to chemotaxis. They're simulated in a grid. You've got biological processes both at the cellular level in terms of cellular activation of macrophages, of T cells, and then you've got the bacteria as well as other rules in 2 dimensions about how the cells can migrate. And at the molecular level, so this is a multi-level model, you've got signaling models. Where those individual agents have rules about the signaling cascades and what gets turned on and turned off. You've got outputs at the population level in terms of total number of bacteria. Here's time post-infection, and about granuloma formation. In this, the infection is contained. You've got a lot of cells and the bacteria in the middle, and here the infection is cleared. Now, this particular paper, which is quite beautiful work, shows you also predictions of what how the various parameter changes will give you changes in, for example, the infected to resting T cell ratio, and controlled growth or uncontrolled growth or clearance of the infection. So one can actually use these models at multi-levels to gain a large amount of information about the system and play with them. So for example, with this type of model, you could make predictions about NF-kappa B signaling. You could use NF-kappa B inhibitors, and your model readout would be both these spatial models, which you could correlate with what you see on histopathology sections, as well as models about various cell populations here, macrophages, T cells, B cells, which you could correlate with flow cytometry. Now, you're probably thinking, wow, that's a hell of a lot of stuff to correlate, which brings up the emphasis on statistical collaboration. You've actually got to have measures of how you're going to compare your granuloma formation here and your histopathology slides and say they're similar, aside from what we fondly call optical fitting, which means I looked at it and it looks really close. Okay. The next concept, which is fairly useful and that we argue about all the time, is some people call it data-driven versus theory-driven models. I like to call them theistic modeling versus non-theistic modeling. Okay, so theistic modeling means that, well, we've got lots of knowledge about how the system works. This is generally how we like to approach these models, right? We're immunologists. We've got this body of evidence that's taken a long time for us to learn, and it's got to be relevant to something. Otherwise, all we are data producing machines. Okay, so we want the modeling to be informed by our knowledge. So when you build those kinds of models, which tend to look like flowcharts or the diagrams that we're very comfortable with, those kinds of models start with what we believe we know about the biology, hence theistic. And the experimental work is designed based on the model, inputs and output measures. And the model parameters are defined by the theoretical model and assumed interactions between all of these elements of the modeling that we think are important. Okay? So what's the obvious issue with these models? Lots and lots of assumptions. Okay? So, you know, we may be missing things, we may be simplifying things too much, we may be dead wrong about our assumptions about how we think things work. So these models may constrain what we find into areas that they may just confirm what we know. And really, you know, it's very nice because it's a lot of work to make a model to have something that's a beautiful picture about what we already know, all right? That really makes us feel good. But models are most useful when they're wrong, because they tell us where we're ignorant, stupid, assume too much, don't know enough, haven't done the right experiment. Okay? And, you know, that being wrong while being painful is what you actually want out of a model. It's what we want out of an experiment anyway. But hopefully wrong for a high reason and not a technical level reason. Okay, non-theistic modeling. This is the kind of modeling that we're really uncomfortable with, right? You take the data. So you've got, you know, RNA-seq from 27 different time points and 2 different conditions. You've got 21,000 genes per time point. You stick the whole thing into a black box. You do the statistical analysis, identify the genes. Then you construct a model based on, not on biological context, but based on what the connections are between the genes, what goes up, what goes down. You know, these models tend to be very large matrices, which represent differential equations or other type of relationships between things. And then all of a sudden, out pops this model, which we hope reflects reality. Okay? So the good thing about that is we get rid of all of our assumptions. The bad thing about that is we get rid of all of our assumptions. Right? Those models are not informed by the biology, and sometimes we come up with correlates, statistical aberrations, other kinds of things. So it helps to recognize the limitations of both of these types of models, and actually sometimes to use both approaches, to go at it from both ways. Okay, so a couple examples of other types of models, then I'm going to lead you through constructing a few immunologic models. So network models to predict gene expression behavior. This is a network model of gene expression and transcription factor interactions that produces that tries to reproduce the various phenotypes of, these are different types of classifications of lymphoma. The particular thing about these models is it allows you to do some perturbations, so reducing BLIMP1 protein stability, NF-kappa B constitutive signaling. You can all think about ways to do these experiments in the laboratory to see if you get this particular phenotype. So these types of network models are very, very useful in some ways, and also fraught with pitfalls. Why? Models require contextual knowledge and they require a focused question. Okay? So here's a little network model. This is a network model of, if you've read the double helix, the story of the discovery of the structure of DNA, these are all the locations that are mentioned in the double helix. Okay? Beautiful network model. Here are our agents. You can recognize Watson and Crick and Rosalind Franklin and Max Delbruck and Linus Pauling. Here's how they are connected in terms of who's mentioned with substantial interactions with who. Does this particular model explain the model that they came up with, the double helix? No. Okay, but it is a beautiful network model. And it does have, the right agents. Okay, so one of the things that we need to keep track of as we're modeling is the model is not reality. Okay, the model is a tool. It's a map. It's something that we use to guide us through the experimental data that is very complex. And Alan Turing, who's really the founder of modern computing and computation, really expressed this quite well. And this is a paper about a mathematical model of morphogenesis based on chemical gradients. And this is really one of my favorite quotes about modeling, okay? In this section, a mathematical model of the growing embryo will be described. This model will be a simplification and an idealization, and consequently a falsification, right? All models are wrong, but are they wrong in an interesting way? Okay? It is hoped that the features retained for discussion are those of greatest importance in the present state of knowledge. Okay? So, this is 1952. So, people have known about this aspect of models, and we have the same uneasiness about models. So one of the things I often get asked when a question that will come up, I'll give a seminar about B-cell modeling, and some of the very good experimentalists will look at me and say, well, you can model anything, right? The model is just this, it's like lies, damn lies, and statistics, okay? And what I hope you'll get from this course is that's not true. Yes, you can model anything, but whether that model actually fits your data is a completely different story if you're honest about it. Okay, so let's build a few immunologic models and talk about the process from the immunologist standpoint of view. Okay, so the first thing is we have to figure out what we want to study. Obviously, we've all got our interests. Then we have to, as Turing said, select the key elements. Okay, these key elements should involve the variables that we can actually measure, the parameters that we think are important, and an abstraction that we think is reasonable for giving us an answer. Okay? Key step, adjust the complexity to what you can actually measure. All right? It's like writing your first manuscript. You want to cram every little piece of knowledge that it took so long to learn into that manuscript, and it reads like a legal defense instead of a scientific paper. Okay? One of the biggest criticisms about models from experimentalists is, but wait, you can't say that. You know, the NK cells do something here, and you're missing all the interferon gamma signaling network. And if you cram all that stuff into a model, but you can't measure it, then you're in trouble. Okay? Your model's not useful. So you've got to start to become comfortable with the idea of an abstraction. We are comfortable with that when we do experiments and when we write our papers, but when we do models, we think, well, you know, we should be able to include much more than we can see. Not true. Okay, so translated into a formal model, which means in a mathematical language or some other type of rule-based representation, design the experiments and select the statistical methods, gather the training data set. Okay, you need 2 data sets for your model. You need one set of data that you train your model with, and you need another separate set of data that you validate your model with. Okay, very important concept. Because otherwise, if you just use your training set, you've got a beautiful representation about what you already know. And of course, it's going to confirm your assumptions and your prejudices. Okay. So you fit the model to the training data, and you know, here's where the part where you bang your head against the wall and smile when it actually works occurs. You adjust the model structure and parameters. Okay, maybe you left something out. You don't have the right parameters. The model structure is wrong. It doesn't work the way it should. And you do all this work with the training data set, and then you generate a model-based prediction. And then you do it all over again. You gather the validation data set, you compare the prediction to the validation data set, you know, hopefully it falls within some realm of what you were hoping to predict, and then you keep doing the whole thing, and eventually you have to exit that infinite loop and publish something. Okay, elements of models, parameters and variables. Okay, variables are things that you measure. Okay. That's the interferon gamma concentration, the expression level of IRF4, the number of activated CD8 T cells of some particular phenotype. Time invariant parameters are quantities that are part of your model. So this is just a linear equation, right? Simple straight line equation. The heavy math will come in a little bit. So these are quantities that don't vary during the time frame of the model. Okay? We know that some of these things, like the rate of proliferation of a B cell after activation, you know, may be constant or may not vary during a particular time frame, but it's not going to be true forever. Okay? So we're confining our model to a particular time frame. Time varying parameters are those that vary over the time frame of the model, and they take a little more mathematics to specify. Okay, differential equations. So this is a very, very simple model. This is a model that Alan Perlson, who will be here talking, developed for HIV. Okay, it violates every sensibility we have as biologists, right, and immunologists. This model has 3 variables: target uninfected T cells, okay, infected T cells, and the HIV virus. Okay? No blood, no tissue, no compartments, no lymph node, no spleen, none of all those other things that we know are really important in the biology. Okay? Differential equations. So this says the change in time, or the change in the number of target cells over time is proportional to the T cell production rate minus the death rate times the number of T cells minus the infection rate. And you notice that for an infection to occur, the T cell and the virus have to come together. Okay, so in a differential equation, when you write one like this, it means that those 2 parameters are stuck, or those 2 variables are combined in that particular expression. And you can write these equations for the virus production rate and the infected cell production rate. And they all have death terms. And one of the things that you'll notice is, so here's the infected T cell rate, and you'll notice that this term here, which is you're subtracting from the uninfected cells the number of infected cells, shows up as a positive term here. Okay, very, very common for differential equation models. So 3 differential equations. You've got virus, you've got a constant viral production rate, and then you've got the infection rate. Okay? How can this simple 3-element model with 5 parameters actually tell you anything useful about HIV infection? Before I answer that, the issues with modeling, okay, make things as simple as possible, but not simpler. This is extra credit to whoever knows who is purported to have said this. Einstein. Although it's probably a paraphrase of a longer, more technical quote from a paper he published. Okay, more parameters and variables. You need more data for model fitting. Okay, the more parameters and variables you have, the more things you have to measure. More variable errors, model variable errors become large. Okay, that's because error for the ultimate output for whatever you're measuring, like the number of infected T cells, if you've got small errors in the measurement of the uninfected T cells, of the virus, of your parameter estimates, all those errors multiply. So your output becomes garbage. It's not that they're additive, they're multiplicative. Okay? Greater risk of unidentifiable parameters, meaning if you have a lot more variables, you may not be able to estimate some of the parameters, or if you have more parameters, you may not be able to estimate those things. Fewer parameters and variables. Okay? You need less data for model fitting. That's a good thing, right? But if the system is overdetermined, you've got many more equations than unknowns, it might not be solvable. And there's the risk that the model may not fit the data. So your model's wrong. It's too simple. So immunologists tend to emphasize this part. Our statistical and modeling colleagues tend to emphasize this part. Yes?
Why is it multiplicative, not additive? Is it because of the inherent nature of ordinary differential equations?
It's because of the nature of, in part, the differential equations, but whenever you have things that are being multiplied together, like for the differential equations, then the error is multiplicative and not additive. And for most of these things, and when you're integrating over time as well. Okay, so how can that very simple model have been useful? Well, this is actually the model fitted to actual data of free virions, infected T cells for HIV. Okay, so the model actually fits the data relatively well over the course of 8 days. But if you go longer than 8 days, the model breaks. Okay, so the model does not fit the data, and what you have are latent infected T cells. So this very simple model identified a key feature of HIV infection in the immunology of that infection that people had not realized. Okay, not because the model was right and fit the data, but because when you tried to extrapolate the model was wrong. The model predicts this part of the curve fairly well, but not this part of the curve. So
does it mean that when you go for a longer period of study, so it goes vary, or whether we should cut short to the shorter one? Or?
It means that your model is likely not going to be applicable to all situations. It will break at some point when you try and extrapolate it. And that's the interesting part. You should always try and break your models, either by going longer period or varying your parameters in some way. So that led to, and here's a more complicated model with virus infection and viral clearance and drugs that came out of that initial model. So very influential model. Let me take you through another example of modeling, and this is one that was motivated by a clinical question. Kidney transplantation. So that's what I do in my other life. So immunologically, we put this foreign thing in, a kidney transplant from somebody else, and the immune system recognizes this as foreign, potentially based on two types of antigens. There's MHC and there are ABO disparate antigens. I'm going to talk mostly about the ABO disparate part. So these ABO antigens have very limited polymorphisms. They're the same ones that are on blood cells. The problem is we make natural antibodies to those. And that's why blood transfusion reactions occur. And what happens is those antibodies are pre-existing, okay? And they bind to the surface of the vascular endothelium. They start a clotting cascade, and basically that kidney, once the surgeons take the clamps off the blood vessels, it gets microthrombi, and the blood vessels clot off, and it turns black on the operating room table. Not such a good thing. So that's why we ABO match most transplants, and we look for antibodies. There is, however, this phenomenon in ABO incompatible types of transplants that after about 6 or 8 weeks, you get this thing called graft accommodation. And that is you get complete inhibition of complement, and although those antibodies may be around, they don't activate complement, and you get accommodation, which means that that transplant will survive. So you've got to get somebody through those initial several weeks of the transplant in order to do an ABO incompatible transplant. Now, the reason for doing that is that these are the types of transplants that you can do right now, but if you were able to get rid of those ABO antibodies and then let them reoccur later, you could actually expand your pool of compatible donors. So, here's the biological cartoon that we would draw as immunologists about antibody kinetics. Antibodies are produced in the bone marrow by long-lived plasma cells. They end up in the bloodstream. If you get B cells that are activated, those cells divide. They differentiate into antibody-secreting cells. Those antibody-secreting cells can be either short-lived or long-lived. The antibodies go into the bloodstream. There's a particular molecular process of IgG recycling, and IgG antibodies are the ones that we care about in this case. And so those antibodies are taken up by a continuous process of pinocytosis. In the vascular endothelial cell, they bind to this neonatal Fc receptor. Antibodies that bind to this Fc receptor are then recycled and released back into the bloodstream. If you saturate or block this receptor, those antibodies are degraded inside the cell. So, when we do one of these ABO incompatible transplants, we use a variety of manipulations that alter those antibodies. So we do something called plasma exchange, where we filter the blood and take out blood proteins, including those antibodies. But, and this is an antibody titer, but those get resynthesized. So we also block the receptor by giving large doses of intravenous immunoglobulin. We give medications, anti-CD20, which kills B cells that might differentiate into plasma cells, and we can give some other medications as well. And we hope that we reach a low enough titer where a transplant is safe, and then we can suppress those titers for a while. Now, what's the problem with this? Okay, this process has never been modeled. So this is about half of the number of protocols in use across the country about how to do this. And I won't go through the various steps, but you can see each of these represents a different element, you know, plasma exchange, intravenous immunoglobulin, plasma exchange plus intravenous immunoglobulin, different medications done at a different time. How are these things designed? Well, they were designed by a bunch of really smart people sitting in a room and saying, I think we should do this every other day. I think this is the mechanism by which this works, so let's try this. These are not lab experiments. They do this to actual people, okay? So the first time that we tried to do an ABO incompatible transplant, this is what happened. We did the plasmapheresis, and these are titers. Here's the safe level for transplant. And we couldn't get those titers down. And there's actually this is done enough so that there's sort of this little cookbook diagram of here's the titer, here's how many plasma exchange treatments you need before, and there's after transplant. So all of a sudden it wasn't cookbook. And this transplant, we didn't actually go ahead and do the transplant, but this failed. We failed to successfully desensitize this person. So I called the guy up who brought this technique to the United States, and I said, Lloyd, have you ever seen this before? And he said, oh yeah, we see this about 15 to 20% of the time. I said, well, is it that they have more plasma cells, they have a higher antibody production rate? He's, I don't know, we never looked. Okay, so what we want to do, the goal of modeling this is predicting successful immune desensitization, and then we want to choose a treatment regimen. Which drugs, the timing and the dose of those things, and we'd want to individualize it to a person. Okay? So the immunology here is getting rid of the antibody and figuring out how to do that. So we simplified the model. Here's the cartoon diagram, intravenous immunoglobulin. Here's apheresis, removal of the antibodies. Here's a compartment that represents all other tissues in the body because this antibody gets exchanged between the vascular space and a tissue space. Bone marrow production of antibody by plasma cells. There's this recycling, and the red rims show you the compartments in the model we designed. Okay? Here's a more abstract cartoon of this model showing you the various rates and things that we needed to either derive from the literature or measure. So this is delta 4-1 is the transport rate of IgG in the tissue back into the vascular space. Here's the administering a bolus of intravenous immunoglobulin. Here's a rate constant for getting rid of that IgG by plasma exchange. Here's the receptor, the IgG binding to the receptor in that endosome, the complex recycling of the IgG and recycling of the receptor. This can then be, so then you write down the list of the things you need to measure and the components of that model, and you see all of these parameters and all of these variables, and how are we going to estimate or measure them? So some of them we can't estimate or measure based on experimental things, but there's data in the literature. So we use that data. So you don't have to be able to estimate all your parameters, but you've got to be able to sort of confine them to some reasonable range. And that can be informed by other experiments. Okay, here are all the variables in that model, but a number of them are hidden variables. We can't really measure the concentration of IgG in the endosomal space. So you're going to need to use math to translate that. There's no way around it. You're going to need to become bilingual. Okay? For many models, you'll have to learn how to read and write a differential equation. You will not have to learn how to solve them. That's what the software is for. Okay? But you will need to recognize the forms of differential equations that can be solved by your software. Okay? The types of software, there are lots of, this should be capacity, there are lots of software packages out there, and this is by no means an exhaustive list for doing this. Okay? Reading a differential equation. Okay? The change in the number of plasma cells over time is equal to the rate of B-cell blast differentiation minus the death rate of plasma cells. Okay? You can write these things. You'll learn how to do this in the course here. This is not that hard, but it does take a little bit of practice. Okay? So here is that beautiful beautiful flowchart diagram described by mathematical equations. All right? I'm not going to go through all of these equations today, but we could sit down for 10 minutes and you could write this after a little bit of practice. It is not that hard. So, what do we know? Okay, and what can we measure? We can measure the kinetics of immunoglobulin. This is a bolus of intravenous immunoglobulin and the blood concentration and the decay phase. And some of you who are familiar with mathematics can tell that this is a two-phase decay of the concentration in the bloodstream. This is plasma exchange, and this is again the concentration of IgG, and you see that it goes down after each treatment, and then there's resynthesis. I'm going to skip over you can estimate single-cell antibody secretion rates, and this is a method that was developed in my laboratory where we did quantitative ELISpots. Each of these, this is a calibration curve. Each of these are beads coated with a known amount of antibody that give a particular spot size, and you can get a standard curve that correlates the femtograms of antibody per spot density or spot intensity. And so we can come up with a statistical estimate of what a single plasma cell secretes in terms of the amount of antibody. So that's a parameter that we can estimate not from measuring blood IgG levels, but you go back into the lab and you get creative. So now let's take the model out for a test drive. Okay, what do I mean by a test drive? Check the boundary conditions. What happens when a parameter is 0 or maximized? What happens when you have no plasma cells? Does your model reflect steady state? If you don't perturb it, is everything a flat line? When does the model blow up? This is the really interesting part. Okay? When can you break the model? You should always try and break your models. Besides being very relevant, it's a lot of fun. Does the model behave like you would expect? The relationship of the parameters, the variable ranges and patterns, and are all of the parameter and variable units correct? So here's a simulation of the model. Remember, a simulation is the numerical implementation of that model. And you can see this is the model fitted to the initial training set of data. This is a bolus of intravenous immunoglobulin. Here's steady state. This is actually compared to another patient. No plasma cells. Well, we didn't really have data to fit this because we couldn't find somebody who had no plasma cells that we could measure IgG levels over. But you can see that the IgG levels go down over time, and that looks pretty reasonable. Here's an agent that destroys plasma cells, not all of them. And you can see that the IgG level goes down, and these are the actual measured data for this patient. Here's plasma exchange, and you can see, well, the fit's pretty good, but maybe not completely good, and we have to figure out what that measure is. So here's more training set data fitting for plasma exchange and for IVIG, and you can see the fits are actually pretty well. This was done good. This was done using DE Discover, and you'll learn how to use this modeling package. So next thing for the test drive is sensitivity analysis. Okay, this involves varying parameters through a biologically meaningful range and looking at the model's sensitivity to any particular parameter, the range of model responses. So this is a sensitivity analysis, and the readout here is the serum concentration of IgG. Here are various parameters in the model, and I'm not going to go through this exhaustively, but you can see that these different lines, the red line is the maximum parameter value, and the blue line is the minimum parameter value. And you can see that, for example, some of the model parameters don't change the model very much. So if you change delta compartment 2 to 3, which is a transport rate of immunoglobulin, the readouts don't change very much. But if you change the production rate of, or the loss, let's see, this is the loss rate of immunoglobulin from the serum, the model is very, very sensitive. So what that tells you is for your model, you're going to have to actually validate it with these kinds of, with the most sensitive parameter estimates, because being off by a little bit is going to change your results a lot. So other types of model errors, the model lacks needed variables or parameters, the relationships are wrong, the model has too many or too few equations, and that they're unidentifiable parameters. So one short example from this is that in this particular model, first we tried to model it with no tissue compartment. So we got rid of this tissue compartment, and you can see the model actually does not fit the data. Okay? So does this mean you can model anything? Well, no. You can't model anything with any particular model. Your model actually has to have the right structure and the right parameters. So when you put back in the tissue compartment, But one of the things we did initially was we didn't limit the saturability of the transport of IgG from the blood into the tissues, and you can see that the model didn't fit again. So we had to put a saturability constant for that, or equation for that particular transport process. And then the model fits pretty well, except here's somebody who got 2 doses of IVIG, so immunoglobulin, and you can see maybe something has changed in the biology because this portion of the model doesn't fit so well anymore. Maybe this neonatal Fc receptor is inducible after you have very high doses of IgG, and so the transport rate increases with the next dose. That wasn't built into our model. But it's not necessarily relevant if we're only modeling one time course. So here's the fitting to a validation set, and we actually did play around a little bit with this second parameter, and you can see 2 different patients. Model fits not so bad. So experimental validation. I can't overemphasize this enough. Yes?
Sorry, I have trouble determining how you can say a model fits or not. So if that particular figure, it doesn't look very, the few points in the bottom are just a little bit moved.
Yep.
And so how do you tell the model fits?
So you need to do some formal fitting statistics, which you're going to learn about in another lecture. I haven't covered here, and actually, if you do the formal fitting statistics, so one thing you can do is just, the least squares difference between what the value the model predicts and your actual measured data. There are more sophisticated methods for doing fitting comparisons, and those measurements will give you sort of the degree of fitness of a particular model. It's the degree of fitness to a particular data set. So if you look at this over a large number of patients for this particular model, those statistics tend to be fairly good, but they're not perfect for each individual. And there are sampling, resampling, and other statistical methods that you can use. So in the interest of time, I'm not going to cover the more quantitative statistical pieces. But you're right. Good statistical methods are needed for those model comparisons. And that means modeling is a team sport, as Hulin mentioned. Okay, you need experimentalists, statisticians, modeler, programmer. Sometimes these people are multi-skilled, so you don't necessarily need 4 people on every project. But you do need all of these skills in the team. The other thing is that you need to design and complete experiments to test your predictions. Okay, so prediction is difficult, especially about the future. Often attributed to Yogi Berra, but apparently this quote is from Niels Bohr. So what we hope to be able to do for this is to actually predict what interventions are going to be needed and in what order for a successful transplant. And this is now a 5-year NIH-funded modeling and immunology grant that came out of that particular question. So very simple, we don't have lots of cells involved. But we were also interested in being able to predict the amount of immunoglobulin after B-cell activation. So this is another abstract part of that bigger picture. And this is a different kind of model. This is an agent-based model where we take B cells and we're simulating an in vitro experiment where we activate them with CpG DNA. They undergo some decisions. Do they get activated or do they die or rest? Once they get activated, do they live or die in each division cycle? So this is a decision. Do they secrete, do they divide? If they divide, that takes a period of time. Do they secrete immunoglobulin? And do they exit this and divide or rest? So this is not a differential equation model. But you'll notice that these things have probabilities. Okay? It's not just a particular rate. And so we designed a set of experiments that measured those or derived those probabilities. So one of the things that often comes up, and this is a question, you get together with a room of modelers and they will ask you, and you will be annoyed, okay? Are you really measuring that? What are you really measuring? No, you're not measuring that. You want me to model that, but you're not measuring that. Okay, and we always have these conversations. So most frequent issue in my experience when constructing a model, often due to differences in language, technical language, and when you're constructing a model, you got to be brutally honest about what you can and are able to measure and are measuring. And sometimes you need to create new experimental and statistical methods to get your parameter estimates. Okay, so for example, in this model we need to measure the rate and time that it takes for cells to divide. Okay, we're not doing video microscopy. Okay, so what are we measuring? We've got these CFSE experiments where you label cells with a dye every time they divide. The concentration of the dye in the cell decreases by 1/2. So you get this nice flow cytometry stuff. Okay? Is that measurement of division time? Absolutely not. But we can use that data to derive statistical distributions for division times and the probability that a cell will divide. So within that larger model, in order to derive parameter estimates, we needed a smaller model. I'm not going to go through the methods for doing this, but suffice to say that we were able to derive division times and probabilities. So here's some of the experimental data we started out with. Here is antibody secretion rates. This is the amount of antibody secreted by a cell, single cell over a 6-hour period as a distribution, and this is number of cells. So these are the things that we could measure in that particular model. These are the model parameters. Note that they're not one value. They are a statistical distribution that is a probability of something happening and the time that probability would represent. So, so 10% of the cells will be able to be activated in about an hour. Or when the cell dies, this is the probability distribution of the disintegration times for a population of cells. Or here's the distribution of the cell division times over a population. So when you do one of these flowchart models and you use a probability distribution, what you're doing is you need to generate a probability for that event occurring in a particular cell. Not all cells will divide with the same amount of time. That really reflects the biology, but you need good statistics in order to do that. And you need to check your model fitting. So this is live cells and dead cells. Here's the CFSE data, and these are the individual division times and the fit. Here are some model readouts, total number of live cells, dead cells, cells awaiting activation, cells awaiting death over the time of the model. And this model was broken initially. Okay, this model assumed, so our readout is IgG secretion per live cells, the proportion of cells secreting IgG. And if we allowed the cells to, once they differentiated into an IgG secreting cell, to start secreting IgG right away, the model was broken. If we made them undergo 2 divisions, which when we went back and looked at our experimental cytometry data, and our ELISpot data, it took 2 divisions for a cell that had intracellular staining for IgG to then start secreting antibody. We needed to build that into the model. When we did that, here's our validation data set, 2 different in vitro experiments, and you can see that the experimental data and the simulated data are almost identical. So a nice broken model. Okay, graphical representation of data. If you come to the symposium, you'll hear a more extensive talk about this. This is key, okay? You know that as an experimentalist and when you're writing a manuscript, you need to communicate what you have done clearly, and the graphics can make or break your paper, okay? You've got beautiful data. You want a beautiful representation of that data and your model, right? So, but that representation of the data has to tell people how you got to that representation. That representation, of course, is an abstraction. Okay, it's a graphical abstraction of your data. It's a spaghetti plot, or it's a heat map, or it's a network diagram. it needs to make sense. And one of the key challenges in modeling and in immunology, the future, is big data. Or data that has summarizing big data in a manuscript, and especially with modeling. So you want to maximize the data to ink ratio. In other words, most of the ink in your plot needs to represent data, not all the annotations. This is from Edward Tufte, who has written a beautiful set of books that's really worth looking at, www.edwardtufte.com, about the graphical representation of data. He's a pioneer in the field. The books are gorgeous and the examples are incredible. You also need to represent the workflow to get the graphic. Graphic should make your audience want to know your story, okay? You know, you look at a paper and you see a beautiful picture and you think, wow, how did they get that? It makes them want to read your paper and it makes them want to read about your model. Learn how to use good graphics tools. Adobe Illustrator, R, Mathematica, MATLAB, there are lots of other plotting packages. That means you're going to have to branch out and learn something more than Excel. Okay, so this is a sensitivity analysis. Now you saw the spaghetti plot, the individual plots of sensitivity. Each of these tiny little boxes here represents 100 simulations. Okay, the tiny boxes. Okay, this is a 15 by 20 grid. So this total figure represents time points it represents approximately 80,000 simulations in one figure. And this shows, using a heat map, how this is simulation time, this is the probability values for these probabilities, and the readout is, total amount of IgG, or total number of IgG secreting cells, total number of IgG producing cells, and red is, or yellow is the highest value. And it tells you, for any particular parameter, how that varies and how the output varies with that parameter. So you can see total number of B cells is not very sensitive to changes in any of these parameters. You can see that the total amount of IgG produced in that experiment is very, very sensitive to the probability of a cell dividing or dying. So, last slide. So this is one of my favorite cartoons by Sid Harris, a scientific cartoonist. And so really what modeling is about is being more explicit in step 2. So what's my dream about how we should talk about models and papers, okay? Now we publish a paper, it's a static thing. Imagine if you publish a paper and not only do you have your data embedded in that paper that somebody else could manipulate, but you've got your model embedded in that paper. So now when somebody says, I think your theory is wrong because, they can say, I think your theory is wrong because if your probability of CD8 T cell activation is half of what it is, here, let me show you by playing with your model, then you get very, very different results, and the structure of your theory has failed to include interactions with NK cells. There are ways of doing that now. There's a format called computational document format, which is put out by the Mathematica people, that allows you to write a manuscript and embed models with sliders that people can manipulate things. There's DE Discover, where you can actually give somebody your entire data set parameters and models, which you guys are going to learn how to use. So I hope I've given you a flavor of how much fun modeling can be as an immunologist, how simple models can address really big problems, and some of the pitfalls at a superficial level and the advantages of using modeling. You're going to learn more about all of these things in the course. This is really a superficial survey. Thank you very much.